very much for having me and organizers and also the really nice talks so far. I hope I don't bore you because it's just the second half of the people get bored. So what I'm going to talk about is leveraging generative AI for generating cells. You know, sounds sci-fi, but so that's basically what we want to do essentially in the next few years. That we want to have a in silico system that people in the lab go and try to see if we want to test cell type A combined with drug B, and there would be a system like a pipeline that would generate these type like cells or single cells for you, right? And before I dive deep into that, maybe for those of you who are not familiar with single cell biology, the idea is that we can now, since 10 years actually, we can isolate cells and then we can read multimodal readouts from those. So for each cell you have gene expression, chromatin accessibility, protein abundance, and also the past few years you can get the spatial location. And ultimately we would like to use all of these modalities to see how the cell change in response to disease or response to perturbations. So our lab and myself, I've worked on multiple directions. So leveraging single cell biology to understand disease and health across patient, zooming into one patient, understanding cellular variability in that patient, and zooming in individual cells in that specific patient to understand how those cells would respond to a certain treatment, and finally having a tissue perspective to see how these cells are talking to each other. So today I'll specifically focus on cell states and perturbations, but we have work also on like patient manifold learning and also tissue biology. So, but I think that for the sake of today, I'll talk about these two now because they are more relevant to drug discovery. So I think that the talk today in the morning highlighted basically a super long pipeline of drug discovery as the first part of that is finding the target. For many of diseases, we don't know the target, and it's not possible to find a target. It's hard. And so the idea is, can we use single cell biology and high resolution readout to find disease phenotypes and potentially narrow down the hypothesis space of cells and targets in this space? And for that, we've been pitching an idea of using reference atlases to analyze single cell data. So reference-based analysis of data is not something new. So we've been using this, I think the field has been using this for since 30 years starting from the reference genome assembly. So there are computational methods that create a reference for the genome, and then we have computational reference mapping for the sequence, right? Because you don't want to reassemble the genome again and again. So you want to map a new data, analyze the similarities and differences across reference and the query genome. Here, the idea that we have here is, can we do the same thing now with cellular biology? Create a reference for each organ, and then using that reference to map new data, and then we use this reference to basically rapidly analyze the query data set and also contextualize the query data set. So these are the series of publications that are focused on the reference mapping side. So computationally, what they allow you to do is, so we assume that there is a method that integrated these reference data sets. So let's say you integrated 30 data sets for long. So you have a neural network that was trained on these 30 data sets. You corrected for the batch effect. And then there is a function f that received this neural network that was trained on the reference data set and a query data set that could be disease. And the task is, without using the reference data, given just the weights for the reference model and the query data set, remove the technical variation between the reference and the query, so you don't have access to the reference. And this has been the pipeline that has been used for cell type annotation, for contextualizing disease, because you can create a healthy map and map disease data. And the idea is that these algorithms have to understand the difference between technical variability and disease. So once you map the query data set, they have to be able to correct for the technical variation between the query and the reference, and then preserve the disease variability. Then as you see in this map, let me see if I can use the marker here. Okay, so as you can see in this map, there are, so the gold parts are actually the query data set. So some parts are aligned with the reference, and there are parts that will not be aligned. And the assumption is the parts that will not be aligned are novel disease states or novel population that are not integrated. They look good, but like, do they actually work in practice? So the first example that I actually showed in one of my papers in my PhD is you can create a healthy reference. In this case, we created a healthy reference across 3 different tissues, so bone marrow, lung, and PBMCs across 400,000 cells. And then we used this algorithm in the previous slide called SCRches to project a query data set from BALF. That was the COVID time, so everyone was just like doing COVID research. I mean, nowadays, maybe it's not sexy anymore. But so you could, so the data set came from 12 patients, so 4 control and 8 patients across moderate and severe COVID. And we trained the reference model, and then we give it to this SCRches approach, and then the query data set was also given. And what you see here is the result of the integration. So part of the query data set was integrated into the reference. So these are the healthy cells from the query. And there are parts that are not well integrated. So at that time, we're asking ourselves whether this is because the model failed. You know, it couldn't correct the variation between the technical variation between the reference and the query, or they are indeed novel disease states that are not present in the reference. And it turned out to be the second case, because specifically we zoomed in into the macrophages because they were bulk of the data. And these macrophages, we saw that the healthy samples from the query were integrated into the healthy macrophages in the reference, and they turned out to be tissue-resident alveolar macrophages. So these are the macrophages that were in the tissue, and they were not the disease phenotype. But the ones that did not integrate, so these big red cluster and also the black cluster here, these are the disease phenotypes. So the red cluster, they are actually monocyte-derived macrophages that are highlighted with CCL2 and CXCL10 because there is an inflammation state here. And the TRAMs, they are highlighted by FAP-P4. So this is the marker for the resident macrophages. So the model was indeed like it could integrate the query into the reference and preserve this novel disease state. And we did the same thing for CD8 T cells. But people have been using this type of computational pipeline have been the core of Human Cell Atlas. So every Human Cell Atlas project that is now shipped, it's shipped with the data, but also a trained model. And there is an interface using these reference mapping algorithms to query that reference. Because in the end, we would like to use these as a map and being able to kind of interactively work with them, right? So this is one example of the first integrated human cell atlas project using existing data sets. To create a human reference for lung. So this is a work led by Lisa Sikima and Malte Luken in Munich, but also in collaboration with I think like 30 different universities and also consortia. This was like more like consortia type of work. And they use this algorithm to create a long atlas across the period of 2 years. So they first created the core reference with 500,000 cells. And they reached out to people. They even like included some novel data, and they extended the core reference by mapping query data set into like 2.2 million cells. And so you can see you have like different lung diseases from carcinoma to IPF and even COVID. And what they showed was, I mean, the same idea that I kind of pitched in a few slides ago, that you can now use this reference to analyze new data set. In this case, they mapped a healthy data set. And they wanted to know whether there are novel cell state, not necessarily disease, could be subpopulation of like healthy samples that might be in the query but not in the reference. Oops. You can look at the paper, but in the end what they showed was like the cells that did not integrate turn out to be subpopulation of the healthy cells that are not in the reference. So people also have been using, these are not my publication anymore, so, but I like the application because it could be quite inspiring for applications of these type of stuff. This is another example that they created a reference for human prefrontal cortex from across the development, from early gestational ages toward adulthood. And this is something that we also work in the lab, that it's observed that some of the gene programs that are activated in disease, you could trace them back to in development too, that there are like similarities in what happens in disease to very early stages in development. And this publication, it's a Cell publication, they showed that by mapping glioblastoma, which is a very lethal brain cancer, into this healthy reference, they could identify the cell state that are happening very early in development. But what they also used this method was to validate their organoid system, because this was an in vivo reference, and now they mapped the organoid data on the top of this in vivo reference to see whether their brain organoids were recapitulating what they would observe in vivo, right? And this is what we're also doing in the lab, that this could help you to find dead-end states or somewhere that your organoid model went wrong, and then go back in the lab and optimize the organoid model to capture the disease or healthy states that you're interested in. Okay. And so I think that was just like a few applications on reference mapping, but this is kind of like the newest work that we have, that now we try to make this more automated and even like applied on patient data. And the challenge to understand what is healthy and disease is, in machine learning, is very similar to the weakly supervised case. So you have the label for the patient, but you don't know the exact labels for every single cell, because the disease would not obviously affect all the cells. And the idea is, can we now create a reference, map new patient data on the top of this reference, and then through classifying the patient, automatically identify what is healthy, what is disease, instead of us having a PhD student go and look at the manifold and see whether there are meaningful states or not. And that's the idea of the project. This is something that I started in my PhD, but now led by another talented PhD student in Munich. And this is, let's see if it, yeah. So the idea is, we have a model called multigrade, and now we call it multi-mill because it's based on multi-instance learning. And so we integrate multimodal data coming from patients. So the architecture is based on a generative model called multigrade, published like I think 2 years ago in ICML, that it integrates across these different modalities, and it gives you this latent space. And what we do is, now we integrate cells into so-called bags. So this is very common in multi-instance learning. And then we give it to a bag classifier. And it's like an attention-based bag classifier. So it basically scores every single cell in that bag to see which cell is more relevant to classify this patient. Ultimately, what you could do with this, you can create a reference across using multiple modalities. You can query this reference using data sets that are not necessarily matching to the reference. So you could have some missing modalities for your new, so let's say if you've created reference with gene expression, chromatin accessibility, now that you have a patient data that only has gene expression, you can still use this method. And it gives you this like out-of-the-box interpretability to find what is healthy, what is diseased. And I won't show it here, but it also gives you a patient manifold that allow you to do some patient similarity. Here's one example that we applied this model to classify patients, so 120 patients in both healthy and IPF. So you can there are different ways to do this type of patient classification. You can obviously use the frequency of the cells that are in healthy and disease. That is very common to do compositional analysis and see the difference. So that's kind of like a baseline here called frequency vector. You can also do pseudobulk. I mean, the question is whether you need single cell at all for this. We showed that you need it. And then you can just kind of like aggregate patients, aggregate cells without really, using an attention function. And then we showed that this actually works better. But in practice, this can automate things, right? So you're searching in an atlas of 2 million cells for the cells that are relevant for disease. And that's the beauty of this, because then, if you look at this, the model so the classification is between IPF and healthy. The model picked up these monocyte-derived macrophages that are present in IPF. And the cells that the model picked up, they actually matched the cells that were characterized by a profibrotic score based on domain knowledge and clinical knowledge that we know about these genes. And you can even get more from this, because the set of genes that you know from the domain knowledge are limited. But now you can look at these cells that the model prioritized and then do some downstream analysis, clustering, and see what are the genes that are driving these cells. And what we show here is we can actually get 16 additional genes that could be added to the profibrotic disease signature. Okay. How much time do I have, Marina? 15 minutes?
12 more minutes.
12 more minutes, okay. So that was the first part that was more focused on prioritizing disease and finding targets, right? Because if you have the healthy map, find the disease, you can go back and see what are the disease cells and potentially can help with the drug, with the target identification. But in the second half, I'll talk about once we identify those targets, and now we have a set of molecules with perturbation, can we use generative AI to predict the response of those cells to the molecules or genetic perturbations that you haven't experimentally profiled? So the idea is that when you do phenotypic drug discovery, you have the phenotype, you don't have the target necessarily. And the idea is how to use these perturbations to search the chemical space to narrow down the hypothesis space of perturbation that could reverse the disease state, right? And it turned out to be, especially if you do high-resolution readouts like single cell, it's a very expensive experiment, especially if you do it at scale. And so you could see a series of work that we pioneered the work of like proposing models that can predict single-cell resolution responses to these perturbations. And that could help to narrow down the hypothesis space, and that could help you to basically do a better experiment design. And this is just one example of like the latest iteration that, I mean, there are now better models that build upon this. So this is called CPA, and I developed this when I was working in Facebook AI. And the idea is what this model does, during the training time, you have cell A perturbed with those two drugs. The model learns to disentangle the drug perturbation and also cell type variation to go to this stem-like cell state, which is free of these cell type and drug perturbation variation. And then at the test time, you can ask the model to generate cells and basically do counterfactual predictions. So you have cell A perturbed with those two drugs, but you haven't, or like cell type A, and you haven't tested cell type A in a cocktail with those three drugs. And that's what the model could give you. It can basically generate whole transcriptome profiles of cells that were perturbed with those three different drugs. So for the sake of time, what this model could give you is, apart from the generative power that you can generate cells, it will give you also a similarity map. This similarity map is inferred based on single cell data. So if the 2 points are close to each other in this map, that means that they had a very similar transcriptomic response. And we actually, in the CPA paper, we used this map to design a second experiment. So somewhere in this map, you have the control. So there is the kind of like the origin. And then the more deviate you from the further you go from the control, that means that drug had an effect in the first place. And so what we did in this example, so this was like A514-9 cell. So the in this scenario, we wanted to use the pre-trained model and then the map that we have to design a second experiment, but with a combinatorial perturbation, so combinatorial drugs. So we used this map and to select 32 mono and combinatorial drug and then fine-tune the model. And so long story short, that we show that if you fine-tune the model is able to predict the combinatorial response of drugs without actually seeing them. But now the next step for us is, can we create these atlases to be more queryable, like as I showed for the cell types. So this is actually unpublished work, hopefully soon on bioRxiv, we call it STMO, that allows you to create a perturbation atlas. So what this model allows you to do, you can integrate across genetic perturbation and chemical perturbations. So there was an animation here, but it's kind of missing. What you get is a cell space that now you correct across different cell lines and different perturbations. So essentially, if two perturbations, no matter whether they are CRISPR-A, I, or drug perturbation, if they have a similar phenotypic effect, they will be embedded close to each other in this map. This is actually not a cell map, but a perturbation map, and we annotated this perturbation map to see what are the mode of action and also the gene programs that these perturbations would induce. And this is in total 7,000 perturbations across genetic perturbation, CRISPR-A, I, and chemical perturbations. And so you can see a UMAP, but then ultimately this could be quite useful because then you can use this for repurposing for many of the downstream tasks in drug discovery. But for us, what was important was can we now use this map to find small molecules that would induce same effect as you do genetic perturbations? Because that you can easily do it in vivo versus the CRISPR perturbation, which is you can do it, but it's hard. And what we show here is an example that you can query a gene expression, you can give it to this SC-MOL model, and then you can ask the model to retrieve drugs, or so in this case, because I'll talk about them, we call it OL drugs. So these are out-of-library drugs. So these are the drugs that you have and measure in single cell. We have in-library drugs. So these are the drugs that you have single-cell measurement. And then you have completely de novo generated drug. So you give it the gene expression profile. The model search the whole space of these perturbations to find the drugs across these three different categories that can, let's see if I can, okay, yeah. So in this example, what we did was there were two CRISPR-A perturbations. This is CBL and then PTPN12. Both of these CRISPR-A activation perturbation would lead cells, so these are K562 cell line, these would lead or induce erythroid lineage. That means that you have genes or you have increase in hemoglobin and also the downstream genes in this pathway. And these are the control cells where there was no CRISPR-A activation for CBL. This is the original CBL activation, so you can see the increase in hemoglobin and also HBZ. And these are the drugs that the model retrieved that would induce the same effect, but also a synthetically generated or de novo generated molecule. So we're testing a bunch of these molecules. I got into this business a year ago and think this whole molecule generation I found out this could be a shaky business. So one has to be quite careful of how we validate this. And so that's why we allowed a system to also retrieve molecules. And then we do the same thing now for PTPN12. So these are all the perturbation and tyrosine kinase inhibitors and even the protein itself. Okay, am I done?
Pretty much.
Okay, can I have 2 more minutes and then the next speaker is not coming, is he?
No, he's here.
Oh, he's here. Okay, so I can see. Okay. Okay, a bunch of these analysis you can actually go beyond gene expression. Now we're doing this on images. So you can have high content imaging. That is the technology that is way cheaper than single cell and many of these high resolution readouts. And the idea is you can have a generative model. The model that I show here is called IMPA, so the preprint is online, is currently in review, and but the revision actually is a completely different paper. But what we show here is that if you give it the control cell, so this just operates on the images, and then you ask the model what will be the response to cytokalasin, which actually causes actin degradation, the model would be able to generate images that would induce this effect. And most importantly, we showed that this model can generalize to unseen compounds. So the compound that it hasn't seen during the training. There is a trade-off here. Obviously, if the compounds are very different from what you've seen in the training, the model would not be able to predict it as accurate as the ones that are similar. And we actually use this model to correct the batch effects in this type of data. Because in larger consortia data, like JOM consortia, they are creating this huge data set across many labs, across different perturbations. And what we show here is that you can use IMPA to correct batch effect and even infer a manifold that I showed you, but now for high content imaging, where you can encode both CRISPR perturbation, also chemical perturbation, and you can actually use this for generation. So I'll just skip this part, but a bunch of these stuff that I showed is just not like academic work. They're actively used in practice. And so our vision is to use generative AI, and that's what I do in my academic lab, that to enable cell engineering. And we recently got a kind of good funding by collaborating through Open Target, we're collaborating with 5 big pharma to develop models for perturbational biology. And we are located in Cambridge, working with University of Cambridge and also Ellis. And so this was work of a lot of people, and we have position and open for collaboration. So if this kind of stuff is interesting to you, please feel free to reach out. Thank you very much.